Hi everyone, thanks for coming to listen to us talk today. So my name is Juliana Maynard. I'm lead scientist for preclinical imaging here at the Medicines Discovery Catapult. And today my colleague Philippa and I are going to talk to you. We want to give you an overview of the work we're doing here at the Medicines Discovery Catapult, who we are and how we're using imaging technologies to visualise drug discovery. So if you have any questions, please do put them in the Q&A box, and we'll look forward to answering them at the end of our webinar. So what is our focus here in Medicines Discovery Catapult? So we're a national centre and we're funded by Innovate UK, and we have a remit to enable and accelerate drug discovery. So we provide unique scientific capabilities and expertise, and we act as a gateway for UK resource. So where we really see the benefits and what is our scientists' focus is that we're building that link between compound to targets and biological effect and translatable systems. And what we're really trying to do is reduce that preclinical attrition of drug discovery projects. So we're translating that clinical information back into the preclinical phase. We're building better models, better biomarkers, and a patient selection strategy. So within our scientific team, we have 5 key areas, and they range from complex cell models to target engagement and validation, biomarkers, bioanalytical technologies, and very relevant to today's webinar is our preclinical imaging team. So within the Medicines Discovery Catapult, we have imaging expertise in multiparametric imaging modalities, and these range from in vivo imaging systems, so positron emission tomography, gamma counting ex vivo analysis at the end of the imaging session, computer tomography, high frequency ultrasound, we've got bioluminescence and near infrared imaging. We have a suite of mass spectrometry imaging, we have DESI and MALDI, our digital spatial profiler, we have the Nanostring DSP, and we also have an advanced microscopy suite, including molecular pathology that we link across all of those techniques. So we have our full home office projects and radiological license. We have access to a wide range of animal models, and we're disease agnostic. We have access to not only commercial radiotracers, but also novel radiochemistry. And excitingly, we have access to also pre-commercial technologies, so technologies that are not commercially available to test your compounds, your delivery methods, we can perform these in-house. And where we really see the benefits of our imaging suite is in that multimodal enhanced interpretation of functional and molecular data. So, imaging drug discovery. Our overarching value proposition, we believe, is to provide visualization of biological functions to accelerate that disease understanding and drug development. And each imaging capability offers a unique solution. And really the projects that we're doing, we always sort of try and compartmentalize them into 5 main key areas. And these really are in looking at organ accumulation, so pharmacokinetics. So when we're injecting a drug into the body, exactly where is it going to, how much is going to the target of interest, and is it going anywhere else? To look at target expression and engagement, so we're injecting the drug into the body, it's getting to the target, but is it actually doing anything when it gets there? Looking at biological activity, so the pharmacodynamics and the optimal biological dose. When we're injecting the drug into the body, is it actually doing anything pathophysiologically? Is it safe to be used? Can we look at toxicities? And also as a surrogate for a clinical endpoint. So are the studies that we're doing preclinically actually characteristic of what we're likely to see in man and for patient benefit? So I'm just going to go through some in vivo imaging examples for you now as to how we have and we can use in vivo imaging. So the first one being in looking at what imaging can do in that drug distribution and drug accumulation space. And here we have examples of both positron emission tomography and also near infrared imaging. So with the zirconium PET distribution image here on the left-hand side, so there are a number of ways in which we can radiolabel a candidate compound, either through direct or indirect labeling. And what we can do is we can measure that distribution and accumulation of the compound across time longitudinally. And certainly, I think in that large molecule and biologic space, this really allows for an opportunity where there currently are no methods to really have a fully quantitative method of understanding the pharmacokinetics and drug accumulation. And zirconium PET distribution, for example, and other radio-labelled versions allows that quantitative method of really understanding where the drug is going to, where it's binding, and how much it's getting there. The second example is here on the right-hand side, and that's with near-infrared imaging. I think recently there's more and more near-infrared dyes becoming available. Near-infrared imaging is great. It allows for really good deep whole body penetration. It's got good signal to noise. We're getting good reproducible data. Fluorescent labelling is potentially easier than some of the PET radio labelling techniques, and so it allows a really good high throughput way of really understanding where our candidate compounds are going to. Also, with the same imaging system, we have bioluminescence imaging, and I think, again, looking to the future, so now the ability to be able to perform lentiviral transduction of luciferase-expressing cell lines and other objects, and what we can really do is have the ability then to look at orthotopic tumour models, and we're going to talk about that in sort of the future methods towards the end of this webinar. But also, you know, in comparing caliper measurements, in tracking tumour volume, it also represents a really good refinement way of, you know, the 3 Rs in our project license as well. So then to look at target engagement, so putting a drug into the body, and actually how do we know it's binding to its target? So this is an example of a study a few years ago using an AstraZeneca compound AZD8186. So this is a PI3 kinase beta inhibitor, and we used F-FDG PET, so fluorodeoxyglucose PET, as a proof of mechanism biomarker. So F-FDG PET is often used as a biomarker with PI3 kinase inhibitors because of the association of the pathway with glucose metabolism. And with this compound in particular, there was a hypothesis that tumours with a PTEN deficiency would benefit from AZD8186 therapy, and that FDG would be a good biomarker. So in the study that we performed, we imaged and we measured after only 2 hours of treatment, so very, very short time after induction of drug. And we did this in 3 models. So in 2 models, they were a PTEN null deficient cell line, and 1 that was PTEN avid. And what we could see is that not only was FDG PET was a good biomarker to assess response and as a proof of mechanism biomarker showing a significant decrease after treatment, but we also confirmed the hypothesis of the project in that we only saw a reduction in those cell lines that were PTEN null, not PTEN avid. Another exciting opportunity that we see is in the use of dynamic FDG PET. So the exact mechanism of why you get increased glycolytic activity in a tumour itself and reduction to therapy is complicated. So it could be as a result of either glut activity, intracellular phosphorylation changes, but also due to tumour oxygenation, blood flow, anti-angiogenic changes, and vessel permeability of the tumour. So by being able to quantify and really compartmentalise what's in compartment 1 and compartment 2 really enables us to understand the mechanism of action of the actual candidate compound itself. So if we look at the diagram here, we can see that as we're injecting FDG into the body, it's entering into that vascular space and it's travelling through then into the interstitial space. It can then either travel into the intracellular space where its mechanism of action, it is trapped within the cell. It does not undergo glycolysis like a normal glucose molecule. But it can also pass back out of the intracellular space into the interstitial space, into the vascular space, and it can do this continuously. So really, as I say, we have a mechanism now of being able to compartmentalise and understand the quantity of FDG that's in the interstitial space with the FDG that's in the intracellular space. So if you look at the graphs on the right-hand side, this is looking at injecting a drug into the body, and we can see no changes in compartment 1. So we're not purely just affecting the blood flow. It's not as a result of a leaky tumour, but we are getting significant changes in compartment 2. So our drug that we've put into the actual animal itself is affecting. So we're looking at proof of mechanism. We're changing either glut transporters, or we're also changing hexokinase activity. So it really gives you an additional, more intricate method of confirming what that mechanism of action of the drug is. Moving on then to assessing whether the drug is actually efficacious, and can we non-invasively determine response to treatment at an early time point. So this is an example here, again using FDG with a PI3 kinase inhibitor, AZD8835. This is a PI3 kinase alpha inhibitor. And we hypothesized that FDG would be a biomarker to determine the minimal effective dose. So here we performed a study at a series of doses, and what we were able to show was that we could see that significant decrease in FDG uptake at doses that were known to be efficacious, but no changes in doses that were not, showing a reduction in tumour volume. So here we're showing that we've got a good biomarker that will be a good clinically relevant biomarker within the clinic, but also that it's a biomarker that could be used to determine early response to treatment, that can be determined if a patient's responding to treatment before we're seeing changes in tumour volume at a later time point. Looking at how we can use imaging to look at toxicity. So is the drug actually safe to be used? And here we've chosen the example of ultrasound looking at cardiac hypertrophy, so enlargement of the heart in cardiac disease. So echocardiography is still the gold standard for being able to assess clinical cardiology. And by being able to track these changes anatomically before pathologically, really it gives us a really good benefit to be able to do this. And by being able to track longitudinal animals, we can make sure we can track this across time. We can also, as you see here on the image on the right hand side, use ultrasound to look at anatomical changes, but also changes in size, but functional changes with the ultrasound itself. And functional changes can often happen before the actual size enlargement. So this really gives us the ability to be able to monitor disease progression, but also if we're seeing cardiac enlargement, it allows for interventional therapy in a toxicity trial. So this is also preclinically as well as clinically relevant. And I think preclinically combining that in vivo imaging with histopathology really gives you that complete assessment of toxicological effects. So my final example before I pass over to my colleague, Philippa, is as a surrogate to predict clinical response. So it's always critical that any preclinical studies we're doing, but if we're going to use and advise to use them in the clinic, then they need to be indicative of actually what's going to happen in the patient's situation. So here we have a study where we're using F-FDG PET preclinically as a biomarker. And what we're doing is we're looking at models that were known not to be effective, so showing no reduction in tumour volume. And to understand if this indeed would translate to what they're seeing with the FDG data. And what we could see was that in those models that were responsive, we were seeing much more of a partial metabolic response than in those models that were not responsive. And in those animals that we're seeing more of a progressive and a stable metabolic response, we're really, that's where they're really sort of sitting. So although the studies are obviously very early, we're really trying to push through that what we're showing preclinically, the levels of reduction that we're looking at is mirroring exactly what we'd hope to see within the patient, using our preclinical studies to guide drug dose, but also imaging time points in the clinic as well. So really providing that really valuable data to feed into those clinical development plans. So I'll pass you over to my colleague now, Philippa, who's going to talk through some of the ex vivo imaging examples and also where we see the future of imaging.
Good morning. So as Juliana says, I'll talk about the ex vivo imaging capabilities here at MDC, but also some of the fixed endpoint analysis that we can do subsequent to some of the in vitro work that we do here at MDC. So first of all, one of the main technologies we're using for ex vivo analysis is mass spectrometry imaging. So there's a variety of different technologies in this space, including things like absorption ionization, TOF-SIMS, so on and so forth, with varying different degrees of resolution, spatial-wise and mass spectrometry-wise. But the two technologies that we have here at MDC are desorption electrospray ionization and matrix-assisted laser desorption ionization, so DESI and MALDI. So the main difference between these two technologies is that with DESI, we're actually applying charged microdroplets of solvents to the surface, and we're desorbing ions in that manner and collecting them into the mass spectrometer. Whereas with MALDI, we apply a matrix, either acidified or otherwise, to the surface of the sample, and we fire a laser at the surface to ablate both the matrix and the analyte, and you then get proton transfer and analyze the resulting ions from that. So there are various different pros and cons between the two technologies, and we'll go into that in a little bit more depth further on in the presentation. But basically what we end up with from mass spectrometry imaging is we're either moving the stage and allowing the stream of charged microdroplets to go across the surface, or the laser to go across the surface. And as we're doing that, we're generating spectra. So here you can see an example of a spectrum. What we end up with at the end is a total spectrum that's averaged across the whole surface area that's been imaged. What we can then do is select one of these peaks that you can see in the spectrum that corresponds to a molecular ion, and see the distribution of that molecular ion throughout the tissue. So this can be anything from a small molecule, so a drug compound, to metabolites, so that could be an endogenous metabolite, or actually a drug metabolite. This can be lipids, peptides, proteins, or even glycans. But one of the most important things about mass spectrometry imaging is that this is an unlabeled approach, so we don't need to label any of these molecules to facilitate this analysis. At the moment, I wouldn't say it's absolutely quantitative, but we can do semi-quantitative work. We can also do relative quantification where an internal standard is applied. So this can be applied to a number of different problems in the drug discovery space. We can do drug distribution type analysis. So in the example that I've got here, you can see that a compound has been applied to some skin. So let's say the first two images on the left are compound A, and the two images to the right, so 3 and 4, are compound B. And here what we've done is taken a section after treating, a cross-section through that skin onto a microscope slide. We've then performed DESI mass spec imaging, and we've been able to identify the drug localization throughout the skin. So you can see the compound A on the left-hand side has actually penetrated quite far down into the tissue. It's actually into the dermal region of the skin, whereas compound B on the right-hand side is sitting on the top of the surface of the skin, so perhaps in the stratum corneum. So to allow us to really correlate that, that presence of the molecule with a given structure in the tissue, we've taken a multimodal approach. Even in just doing H&E staining of the section subsequent to analysis, because DESI is actually non-destructive. And we've been able to overlay the mass spectrometry image on top of that. And then the images in space 2 and 4 are actually overlaid onto 2 endogenous molecules that you can see there in red and blue, just to give some context to what we're looking at. So in addition to doing the DESI analysis here, we went a step further, because although we believe that the drug to be sitting on the top of the surface in the example on the right hand side, we wanted to know, was it actually just in the stratum corneum, or was it going into the epidermis? Obviously this is quite important for this particular compound that we were looking at, and in order to do that we switched to using MALDI. So the main reason for this being that MALDI provides an extra level of spatial resolution at the moment compared to DESI. So what we were able to do then is really genuinely confirm that the compound, in the case of compound B, was actually sitting on the stratum corneum and not penetrating any further into the skin. So here we're actually able to look vaguely at target engagement and check whether or not a compound is going to where we think it is in the sample. So in addition to looking at ex vivo tissue, we're obviously trying to humanize our approach to drug discovery as much as possible, to have better prediction of what we're likely to see in the clinic. So one of the ways that we're doing this is to look at using spheroids or organoid models. So from iPSC-derived cells, for example, or primary cells. And in this case here, you can just see an example of a kidney organoid that's been grown up. This is actually one containing at least 12 separate different kidney cell types. So the images that you can see here have been generated as a result of immunofluorescence imaging using confocal microscope. So what my colleague Lorna Hale has been able to do here is to use that immunofluorescence imaging to actually highlight some of the defined structures within this kidney organoid. And what this means ultimately is that if we were to do a drug distribution study, or if we were to look for biomarkers here, we'd be able to identify where they were sitting within the structure itself. So to take this further, we've actually been doing some work on imaging of these organoids, so taking a section through these organoids and mounting them onto a glass slide and performing MALDI imaging. So the examples that you can see here are fairly rudimentary at the moment. So here you can see just the distribution of endogenous lipids. And here on the left-hand side, you can see in red and green the distribution of two different lipids that are localized clearly in different regions of the tissue. And it's our hope that we can correlate this with the immunofluorescence imaging going forward to be able to identify where different compounds or different endogenous molecules are sitting within the various structures of that kidney organoid. So again, we've really to use the extra power of resolution that we get with MALDI imaging and to push it even further down to the 10 micron image that you can see on the right hand side. And here you can see there's 3 different colored regions in the image, and these correspond to 3 different molecules present within that tissue section. And you can see again that we're potentially highlighting much more features within the tissue section there. So in addition to looking at single molecule distribution within a section within a tissue, we can also profile a tissue, either to look for different responses within a tissue to a given therapeutic, or to actually classify different regions of tissue that might be of interest. So in the center, that's what you can see, a segmentation map that results from doing k-means clustering analysis to highlight different regions within that kidney organoid, possibly correlating to different anatomical features. So in addition to the mass spectrometry imaging, we also have an advanced microscopy suite. And this is just one example of some data that's come from that. So here, one of my colleagues has been looking at super resolution imaging. And here, what you can see lighting up in each of these images is actually the location of a given receptor, so EGF receptor. So this is normally present in cell membranes. And if we look at the image, the second image on the left, you can see that there are clearly quite a large number of receptors there. But as we improve the resolution, we can get an even better idea of how many receptors are present within that cell feature. So the interesting thing about this technology is that we can actually gain so much more resolution that we can look at distribution of receptors or compounds actually intracellularly. So even down to looking at their presence in defined structures such as mitochondria, for example. And this is really useful for target validation and target engagement studies to really get that extra level of confirmation of where we're engaging our target with a given therapeutic. So moving on, we also recently have started working with Nanostring on their digital spatial profiler. So my colleague Gail Marshall does a lot of work with Nanostring, and she's been looking at immuno-oncology profiling. So just to give some background on the Nanostring digital spatial profiler, it's a technology that's not yet fully released in the UK, but we will be one of the first 4 sites to get hold of this technology in the UK. And basically what we can do with Nanostring is we can actually quantitatively, spatially analyze up to 800 protein or RNA targets within a given tissue section. So at the moment, this is currently based working on FFP tissue. However, we believe that we should be able to do this also on fresh frozen tissue, depending on the sample type that people may have. So this can be done on clinical samples. As with any of the other technologies, we can implement them on clinical samples from biobanks, as well as looking at taking some of the ex vivo tissue from some of the experiments being done in the preclinical imaging suite. So what you can see here is an example where the tissue has been stained, first of all with 4 antibodies to look for morphological features. And it's then been split, as you can see in the contour map, into 12 different regions, where they've been looking at macrophage activity, so macrophage localization. And then what you can see on the right-hand side in the heat map is that using Nanostring DSP, we've been able to look at how much of a given protein, each of the given proteins has been expressed within each of those 12 regions. So to show another example from Nanostring, because in addition to being able to look at up to 800 proteins, we can also look at RNA. So in this case, you can see a colorectal cancer section. And again, it's been first looked at for morphology by applying for initial antibodies to the tissue. So we can see different morphological features in there correlating to stroma CD3 enriched regions and tumor region. And here, a few discrete locations have been extracted and then looked at for RNA expression within each of those different regions. So one of the most interesting things about Nanostring is that it is fully quantitative, this DSP technology. So you can analyze down to even single copies of a protein or RNA within a tissue section. So this is very, very useful for biomarker studies to really get an idea of whether or not the drug is having the effect that we expect it to be having on a given tissue. So finally, moving forward, we believe that there are a number of future opportunities that MDC would like to be involved in and that we are taking forward. So one of these is that my colleague Juliana Maynard is looking at zirconium cell labeling as an approach for PET imaging. So in this particular case, Juliana is looking at implementing this approach to be able to track immune cells throughout the living system, so throughout a mouse, for example, or a rat. And this is quite important, for example, in studies where you might be looking at seeing whether or not lymphocytes or T cells or B cells are actually infiltrating into a tumor environment. And this may be either in response to having treated with chemotherapy or radiotherapy, for example, or any given therapeutic. So interestingly, this is an approach that we're hoping to take through from not only in vivo imaging, but also through to some of the ex vivo imaging. So we're also collaborating to be able to take a similar approach in mass spectrometry imaging. So being able to identify populations of different cells within a different tissue section. So where we are seeing upregulation or downregulation of a biomarker, or seeing localization of a particular therapeutic or its metabolites, we can also correlate that, hopefully in the future, to a given cell type within the tissue to give us that much more biological relevance to what we're looking at. So finally, as I've mentioned previously, through looking at some of our organoid models or spheroid models, it's also really important that we continue to sort of humanize our approach, even to some of the in vivo experiments that we're doing. So obviously we can do things like looking at xenograft samples, taking human cells and putting them subcutaneously into a mouse model. But also Juliana is doing a lot of work to be able to look at things like orthotopic tumor models. So here you have those xenografts really in the environment that's much more predictive of where it would be in a real clinical example. To be able to look really a bit and get a bit more biological relevance to what we're looking at here. And again, this is something that we'll be using for in vivo imaging, but also then obviously being able to leverage the other imaging technologies that we have to analyze those samples and get that much more data in models that are really predictive of what we expect to see in the clinic. So finally, just to summarize, we've got a number of different multimodal pipelines that we work with. Obviously, this can be split up for simplicity into two streams. So we can do in vivo real-time imaging using technologies such as PET-CT, ultrasound, near infrared imaging, and bioluminescence imaging. We can then dissect and take sections through tissue to perform ex vivo imaging using techniques such as mass spectrometry, the digital spatial profiler for spatial transcriptomics, and also advanced microscopy. In addition to that, we are also working at MDC with a lot of in vitro experimentation, so being able to do live real-time imaging in vitro using things even such as technology as the IncuSite, which is an incubator that allows us to actually image on the fly whilst our experiments are ongoing and cells are growing, in addition to confocal microscopy. And then to take that through to fixed endpoint imaging, so where we may take terminal cell populations or sections through, for example, complex cells like cell models like organoids or spheroids, and then perform mass spectrometry imaging and spatial transcriptomics and advanced microscopy on those. So obviously this is all very nice. We can do a large amount of imaging, multimodal imaging in various different scales, either live or ex vivo after the fact. But the most important thing is that we need to really choose the right approach to answer any given biological question. So for example, if we're looking at a biomarker study and somebody comes to us wanting to do some biomarker analysis, it's really important that we think about whether or not that analysis is targeted. Do we know what biomarker we're looking for? Is it untargeted and exploratory? So are we going for more of a biomarker discovery process? Does it need to be quantitative, for example, or does it need to be correlated with the given localization of a drug or a drug's metabolites? So by thinking about these things, we're really able to direct our approach in the appropriate manner to the various different technologies that we have available at hand. And one of the main future challenges that we really see of working in this multimodal, multiscale way is allowing for that dynamic integration of imaging data. So with the current software and the current technology that we have, we're able to take static images from each approach and overlay the data from each approach upon each other. But at the moment, we're only able to keep the actual dynamic function of any one technology at any one time. So going forward, we think one of the things that we'd like to pose to the community is how we might be able to dynamically integrate that data so that we can manipulate all of it at the same time to really reach full potential. So we'd just like to acknowledge some of our colleagues here at MDC. So some of the main ones being Gail Marshall for the digital spatial profiler imaging, Isabel, for example, for handling our advanced microscopy suite, Matthew Burnham for the super resolution imaging, and the whole of the discovery science and technology department here at the Medicines Discovery Catapult. Thank you very much for taking the time to listen to us today.